we said the calcium start muscle contraction. Now we want to know where is calcium stored in the muscle cell. The calcium is stored in a well-developed organelle within the muscle cell called sarcoplasmic reticulum. All of the calcium is sequestrated within these organelles. And when these cells are stimulated by a nerve through the synapse, this organelle start to release the calcium into the sarcoplasm and cause the muscle contractions. But here you can see again and the muscle innervated by a motor neuron, a site of synapse. After releasing the neurotransmitter into the synaptic cleft, and the neurotransmitter start action potential or depolarisation in the cell, in the membrane of muscle cell. Like this, you can see a start action potential from here, and this action potential, actually when we magnify this part, the action potential in the cell membrane actually open the voltage gated calcium channel and open these channels and cause influx of calcium from the extracellular fluid into the sarcoplasms. This calcium open another calcium channels on the membrane of sarcoplasmic reticulum. After opening this channel, calcium released into the sarcoplasm and cause contractions. When the depolarization stops by a nerve, this calcium sequestrated by a pump on the again membrane of sarcoplasmic reticulum into the sarcoplasmic reticulum, removed from sarcoplasmic removed from the cytoplasm of muscle and stop muscle contractions. You see in this picture the sarcoplasmic reticulum surround all muscle myofibrils, and all parts attached together actually make a one organelles within the muscle cells. But these organelles have one problem, and the membrane of these organelles cannot produce action potential. That means when the cell membrane of muscle produce action potential and reach to the sarcoplasmic reticulum membrane near to this membrane, this action potential from the cell cannot produce any action potential in the membrane of sarcoplasmic reticulum. And from the other side, we need to calcium release from the sarcoplasmic reticulum throughout the muscle fiber at the same time to produce uniform contraction of all myofibrils at the same time. To solve this problem, we need to bring the cell membrane with action potential property into the cytoplasm. You can see the sarcolemmal or cell membrane make a long finger-like invagination within the sarcoplasms. These structures called transverse tubules or T-tubule penetrate deeply and surround all myofibrils like a sarcoplasmic reticulum, and brings action potential in all over part of sarcoplasms, and cause releasing of calcium throughout all sarcoplasmic reticulum, and cause the uniform contraction of all myofibrils. This structure is said called the transverse tubules or T tubules. You can see here, when the action potential started at the sarcolemma by a neurotransmitter from a motor neurons, the action potential through these transverse tubules reach to the sarcoplasmic reticulum and release the calcium from this organelle. These tubules called transverse tubule systems or T tubules. Again, here you can see invagination of cell membrane and surrounding all myofibrils, and this cell membrane like electrical wire come deeply into the sarcoplasm and trigger releasing of calcium from the sarcoplasmic reticulum. In response, sarcoplasmic reticulum also produce a large system, a large tubules near these transverse or T tubules to sequester more calcium near to the T tubules. And at the time of an action potential, they can release a lot of calcium into the cytoplasm and cause muscle contractions. Again, here you can see sarcolemma, myofibrils, sarcoplasmic reticulum surround all myofibrils, mitochondria, and peripheral position of the nucleus. Here you can see, in this part you can see cross section. Here you can see sarcoplasmic reticulum, transverse tubules, and near to the transverse tubule we have a large cistern of sarcoplasmic reticulum called terminal cistern. Terminal cistern. In the both sides of a transverse tubule we have two terminal cisterns. The transverse tubules round between the myofibrils, or on the surface of myofibrils, at the site or at the boundary of I band and A band. You can see here is A band, this part is I band. At the boundary of these two bands, the transverse tubule run deeply into the muscle cell. On both sides also we have a terminal cistern. So we can say each sarcomeres of the skeletal muscles has two transverse tubules on both sides of A band. In the TEM pictures, transmission electron microscope pictures, when we have longitudinal sections from the muscle, skeletal muscle or muscle fibers, we see intermittent three tubes close to each other. You can see here in this picture, we can see three tubes with equal distance. These three tubules within the skeletal muscle called triad. Actually, one transverse tubules and two terminal cisterns produce triad within the skeletal muscle. Here again you can see transverse tubules encircling around the muscle fibers, terminal cistern around each transverse tubules, triad. Usually in the TM we can see a structure like this, repetition of these structures, or triad. Again here, transverse tubule, so the depolarization of action potential through the cell membrane of sarcolemmal reach to the transverse tubules, and you can see open the voltage gated calcium channels and influx of calcium and releasing calcium from the sarcoplasmic reticulum into the cytoplasm and creation muscle contraction. In this slide you can see we have three motor neurons originated from the anterior horn of spinal cord. You can see the purple one innervated 3 muscle fiber, the red one again innervated 3 muscle fibers, but the blue one you can see innervated 4 muscle fibers. When the motor neurons innervated a few number of muscle fibers, for example here you can see 1 motor neurons innervated 1 muscle fiber. This create precise contraction or precise activity in that muscles. Like for example extraocular muscle for the eye movement, or the finger muscle. You can, for example, take or pick up object from the ground by your finger very precisely. But when the muscle, the motor neuron innervated a large number of the muscle fibers, this create a coarse movement. For example, like muscle of back muscle, they are striated, our back, called erector spinal muscle. A motor neuron plus all of the muscle fiber is controlled, called motor unit. So we can say we have 3 motor units here. Motor unit with a few muscle fiber create precise movement, and motor unit with a lot of muscle fibers create coarse movement. As real picture you can see branching of axon and innervation of several muscle fiber, and you can see the end of each axon, terminal bottom, synapse with a cell membrane of muscle fiber. We magnify here the end of axon or terminal bottom. The axon at the end loses its myelin sheath, just covered by a glial cell, Schwann cell, and make a synapse with a sarcolemma. The sarcolemma at the site of synapse, you can see make some folding to increase the surface. Why? Because this cell membrane here should provide enough receptor for the neurotransmitter. Usually the neurotransmitter in the muscle is acetylcholine, so the postsynaptic membrane or the muscle cell membrane should provide enough space for a lot of number of the receptor for the acetylcholine. This part, or the synapse between the nerve and muscle, called motor end plate or myoneural junction or neuromuscular junction. All of these names for this part of synapses between the nerve and muscle. You can see here motor end plate here releasing the neurotransmitter, usually the acetylcholine, from the nerve, and you can see the receptor of these neurotransmitter on the sarcolemma. Usually this receptor is a kind of ion channel, usually the sodium channels. By attaching the neurotransmitter to these channels, is open and cause influx of sodium and cause depolarization. Let's see one of muscle disorder called myasthenia gravis. Actually, the myasthenia gravis is a autonomic disorder. In this disorder, the autoimmune system starts to produce antibody against the acetylcholine receptors. You can see the antibody attached to the acetylcholine receptor and interferes attaching of neurotransmitters and cause the periodical muscle weakness. The muscle cell, to solve this problem, endocytose the affected receptors and make a new receptors, but again this receptor occupied by an antibody, and all of these attachment cause weakness of the muscle. The first muscle affected by this disorder commonly is the eyelid muscle. You can see cause ptosis or dropping of eyelids. The first sign of myasthenia gravis. In the skeletal muscle, in addition to motor and neurons, we have some sensory neurons, and we have some sensory organelles within the muscle. These organelles called encapsulated proprioceptors. We have two types of these receptors in the skeletal muscle. One of them within the muscle, and another within its tendon. Within the muscle called muscle spindle, and in the tendon called Golgi tendon organ. Muscle spindle actually is a modified fascicle with a special proprioceptor, with a special premesenteric, a connective tissue encapsulate the fascicle. In the cross section, TEM and also the light microscope, we can see modified or a special premesenteric around this modified fascicle. Within the fascicle, actually the space filled with a fluid, and within this fluid we have some modified muscle fiber, usually the small in diameter compared to the other muscle fiber. This muscle fiber within the muscle spindle called intrafusal muscle fiber, and the other muscle fiber called extrafusal within the muscle spindle, and extrafusal muscle fiber. You can see the sensory fiber penetrate into the muscle spindle and wrap around the modified intrafusal muscle fiber. The sensory organ actually detect the stretch of muscle, the rate of stretch in the muscle during the muscle stretching. Within the tendon, we have a Golgi tendon organ. It's also a kind of stretch detector, but within the tendon. I will explain with more detail in next slides. Here you can see again the muscle spindle, capsule or modified premesenteric, intrafusal, and sensory fiber. You can see a wrap around this intrafusal muscle fiber. Again, here you can see tendon organ or Golgi tendon organ. When we stretch the muscle, you can see the number of the signals relayed to the spinal cord increased, or the action potential frequency increased during the stretch by stretching the muscle. And during the muscle contraction, number of these action potential decreased. So when we stretch each muscle, actually we stimuli the muscle spinal. Let's see a good example for better understanding the function of muscle spinal. You can see here in our legs and on our thigh, we have two kind of muscles in our limbs. For example, in the foot, you can see in the anterior part, we have extensor muscle called quadriceps, and in the back of the thigh, we have a flexor muscle or a hamstring muscle. In the medical examination, we have a test called patellar reflex or knee jerk for examination of muscle functions. A patient or a person sit on a chair and hang his foot or her foot, and with a test hammer tapping on the patellar tendon immediately under the kneecap or patella, we tap in this part, and this tapping causes bending of this tendon and stretching the quadriceps muscles. The rate of this stretch detected by a muscle spindle and relay this information to the spinal cord. Within the spinal cord, the sensory nerve stimulated two types of motor neurons. You can see the first one comes to the quadriceps muscle, the second one comes to the opposite muscle or flexor. The first one, actually, you can see here, plus means cause contraction of this muscle, at the same time relax the opposite muscle. So the muscle spindle by this way regulate the function of opposite muscle. We need this kind of function during the running, during the walking. We cannot have the contraction of both opposite muscle. With contraction of both of them, we cannot walk, we cannot run. So by this way, muscle spindle regulate the function of opposite muscle. When we tap on this tendon by stretching, at the end we have a kicking movement of the leg. About the Golgi tendon organ, you can see here, instead of the muscle fiber here in the tendon, we have a collagen fibers. So the sensory nerve run between these collagen fibers, and during the tendon stretch, we have during the muscle contractions, this stretch actually stimuli the muscle fibers, or stimulate the sensory nerve, and this information against or this stretch detected by this sensory nerve. During the tendon stretch, I said we have at the time of muscle contractions, the rate of this stretching relayed by a sensory nerve to the spinal cord. Like a muscle spindle, you can see again we have a stimulation of two motor neurons. One of them comes for the contracted muscle, causes stretching of tendon, and another for the opposite muscle. But here it's vice versa. You can see this stretching cause relaxing of the contracted muscle and cause the contraction of the opposite muscle. So by this way, the Golgi tendon organs decrease the stretching of tendon and prevent tendon tearing or rupturing by this way, especially during the lifting of high weight, heavy during the lifting the heavy weight. About the skeletal muscle fiber types. We have different types of muscle fiber. Under the light microscope, we see same muscle fibers by a usual tissue processing, I mean the Cheney staining. But in the function, we see different type of function. For example, the extraocular muscles have a very quick or rapid contractions, but in the back muscle or erector spinal muscles, they have a very slow contractions for a longer period without fatigue. All of this information indicate we have a different type of muscles. Actually, this type of muscle, they have a different expression of the contractility proteins, regulatory proteins, and the protein affecting the oxygen storage. So the cells have a different content. According to these different content, we have a different type of muscle fibers. When we stained the muscle cell with a spatial staining, you can see we have a different type of colors. Some of them are white, some of them are dark, and some of them are intermediate. It means they have a different content. For example, different content of enzyme, different content of the proteins within the cytoplasm. We can subdivide the muscle fibers according to two bases. The first one is according to the rate of contraction. According to the rate of contraction, we have a slow and fast muscle fiber. A slow contraction, fast contraction. And the second one, according to the main pathway of ATP synthesis. You know, we have two pathways for producing ATP. Oxidative phosphorylation by consuming oxygen, and without oxygen or in the low oxygen, we have an anaerobic glycolysis. So according to this main pathway, we have oxidative fibers and glycolytic fibers. The slow oxidative fibers, also called type 1 or red fibers, in the fresh section from these muscles, or the fresh color or appearance of this muscle is red, so we call them red muscle. And the fast glycolytic fibers called white. This white again is because of the fresh color of this muscle, or type 2B. And the third type of muscle actually is intermediate fibers. They have a fast oxidative glycolytic fibers. Some books, instead of the saying fast, called intermediate oxidative glycolytic fibers, or type 2A. Why they are different, you can see in this table. The type 1 fibers, or the red fiber, or the fast fiber, the slow fiber, slow type 1 fibers or red fibers, We said they have a contraction for longer times without fatigue. Why they don't have a fatigue? Because they have a huge source of oxygen within the cells. They can supply good for oxygen. Why? Because they have a lot of mitochondria. They have a high density of the capillary, and they have a lot of number of the myoglobin within the cytoplasm. So by these three components, they can provide high amount of oxygen. So they go the pathway of oxidative phosphorylations to producing ATP for muscle contraction. So they can contract for a longer time without fatigue. But in the third column, you can see the white or type 2b. We see a sparse mitochondria, sparse capillary, low density of capillary, and low density of myoglobin. So they cannot prepare enough oxygen for producing ATP. So the cells go the anaerobic pathway, glycolysis, and you know in the glycolysis pathway or glycolytic pathway, in addition to the cell produce ATP, we have a byproduct or side product called lactic acid. And during producing the ATP, the lactic acid also produce and accumulate within the sarcoplasm. And high amount of lactic acid causes fatigue and prevent muscle contraction. And these kind of muscles for contraction need a period of recovery to get rid of the lactic acid. And you can see the intermediate fibers here between two muscle fibers. And also I have an example for them. For example, you can see back muscle or postural muscle of backs, major muscle of the legs are intermediate, and extraocular muscles are white muscles. They get fatigue very fast because producing a lot of lactic acid during the glycolysis. In our body, we don't have a pure muscle from fiber type 1 or type 2b or type 2a. Usually all of our muscle are mix of them. But for example, extraocular muscle, they have a lot of fibers type 2b compared to the other fibers. We said we have a myoglobin within the sarcoplasm. Actually, the myoglobin is kind of protein that this protein conjugated with a non-protein structure called hem. And hem, you know, have a iron within this structure of hem, and iron can attach to the oxygen, and they can storage the oxygen, or they can store the oxygen within the cytoplasm and supply oxygen for a muscle contractions. This is very similar to the hemoglobin within the red blood cell. High amount of myoglobin supply a high amount of oxygen for a muscle contractions.